Hi, my name is Morgan Yonke, and this is Cell Biology, and this is my molecule lab. Here is my concept map. I'm going to just show it to you up close really quick before I start explaining. I have two different separate parts of the map. There's the first part, cell layers, and then the second part, cell models. So I'm going to start with the cell layers. And I started with cell layers, and I put three different branches out from cell layers: membrane protein, plasma membrane, phospholipid bilayer. From the next one I put into vesicles. From vesicles I put membrane channels, gated ion channels, and off to the side selected permeability, just because some vesicles can be selectively permeable, others aren't. So I just put that off to the side, not necessarily with these, but off to the side from vesicles. From gated ion channels, I put electrochemical gradient and concentrate gradient. From concentrate gradient goes hypo, iso, and hyper solutions. And then from electrochemical gradient and concentrate gradient comes all of these: osmosis, passive transport, simple diffusion, active transport. And then from passive, channel-mediated passive transport. And then from the membrane proteins, I have listed all the membrane proteins, which would be integral membrane proteins, peripheral membrane proteins, proteins attached, transporter membrane proteins, anchor membrane proteins, receptor membrane proteins, enzyme membrane proteins, monolayer, lipid linked. And those are all the different types of membrane proteins in this particular lab. And then on to my next concept map would be cell models. And so I started off with the two separate cell models. One is the sandwich model, which is by Dason and Danelli. And that model describes a phospholipid bilayer that lies between two layers of globular proteins. And then the next one would be the fluid mosaic model by Singer and Nicholson, which also explains various observations regarding the functional cell membranes. It describes a two-dimensional liquid in which phospholipid and protein molecules diffuse easily. And so those are those two models. And then from those two models, I put some of the molecules: the amphithetic molecule and the detergent molecule, and then glycoprotein and glycolipid. From glycolipid, I put saturated and unsaturated fatty acids, and then just regular lipids. And then I put from glycoprotein, proteoglycans, and sterols. And then detergent molecules alone, and then amphithetic molecule, I have the hydrophilic heads and the hydrophobic tails. And so thank you guys for watching my video, and I appreciate your time, and I hope you all enjoyed. And I'll get another close-up of the cell models so that you can see.